Hello everyone, this is Akshay Trimukhe. Today we are going to start with an important topic of dental histology, that is amelogenesis. So what is amelogenesis? Or define amelogenesis. Amelogenesis is nothing but a physiological process of formation and maturation of enamel. So amelogenesis is basically formation and maturation of enamel. So that is amelogenesis. Now we are going to start with our whiteboard lecture. So formation of enamel is basically done by your which cells? Ameloblast cells. So ameloblast cells are the former of your enamel. Like we'll discuss other also, like dentin is formed by your odontoblast cells. Cementum is formed by your cementoblast cells. Okay, so we have just defined our process of amelogenesis. Now we are going to see in detail amelogenesis. Now amelogenesis starts at your early bell stage, or you can say early crown stage. Early bell stage of your tooth development. Now let us see how the early bell stage looks like. When you see early bell stage, you will differentiate some structures in this, or you will say differentiate some type of tissues or layers of cells. So okay, this is your the adjacent layer which I'm highlighting now with my marker. This is your dental follicle. The internal part now which I'm highlighting is your dental papillae. This part or the inner layer which is adjacent to dental papillae is called as your inner enamel epithelium. Now which I am making. This is called as your inner enamel epithelium. I'm not going to make this entirely. This is your inner enamel epithelium with its nuclei. Okay. This is your basement membrane which separates the inner enamel epithelium from your dental papillae. Now this part which is adjacent to your dental follicle or dental sac, that is called as your external enamel epithelium. Again, this I am not going to make it entirely. So this is your external enamel epithelium, internal enamel epithelium. Now between these two layers of cell, there are again two layers which are, first is, that is your stellate reticulum. They are star shaped. They have again nutritive and they work as shock absorbers and protect the developing tooth underlying. Okay, so this is your stellate reticulum, and there are one more layer of cell. Means actually this layer is made up of 2 to 3 layers. It is called as a stratum intermedium. Now note, stratum intermedium is just found at your coronal part, so that's why it is very important, because this stratum intermedium has a highly alkaline phosphatase activity, and contains alkaline phosphatase enzyme, which is in turn giving or boosting this enamel cells or ameloblast cells to form or secrete what you can say enamel, or in the process of amelogenesis. So this is your stratum intermedium. As well, it also contains some glycogen or glycogen content, because during the first, or you can say, formation of enamel, or just what you can say, when the first layer of dentin is been laid down by the odontoblast cell, this layer of cells or the pre-ameloblasts which have not yet been differentiated into ameloblast, and before it secretes enamel, it needs some energy or nutritive supply, and this nutritive supply was previously gained from your dental papillae, because these cells are now alive, so it will be requiring some nutrition. This nutrition was first derived from the dental papillae, because dental papillae was supplied by capillaries. But as the dentin is been formed, or first layer of dentin is formed, this supply has been cut off. So this ameloblast cell is now in a big question, that from where they will derive its nutrition. So before these all stages, this stratum intermedium is starts storing glycogen, and this glycogen is being used up by the ameloblast cells, and it can undergo the starting stages of amelogenesis. Later on, this what you can say external enamel epithelium collapses, and the adjacent dental follicle cells which were having the capillaries, it invaginates and they makes contact with this, and they derives its nutrition from the external this capillaries, which is actually from the dental follicle area. So this was something which happens in your advanced bell stage, that is later part. So this was something we have discussed in early bell stage. So basically, amelogenesis starts at your early bell stage, or your early crown stage. Now amelogenesis is divided into two part, or amelogenesis is like, first it will undergo two steps, that is, first is your secretory step and second is your maturative step. Because amelogenesis, first it will secrete the enamel matrix. That enamel matrix is completely, or you can say majorly, or containing organic matrix. It doesn't have much of hydroxyapatite crystals. It is just 30% of mineral content or calcium content or hydroxyapatite crystals. So it is just 30% mineralized. And later on, it second step is your maturative step. In maturation stage, what happens is that this organic material is being extruded out, or it is been effluxed out by some means that we'll discuss later on, and thus the enamel rods which were laid down, it attains its size, height, width, or whatever the parameters, and it becomes completely mineralized, that is 100%. And we know enamel is 96% mineralized or contains inorganic matter, inorganic content, and 4% organic and water content. So it becomes 96% mineralized. This not 100%, that is 96 mineralized, because it contains some traces of organic as well as water content. So this just, it was a short idea ki amelogenesis will involve two steps, that is secretory and maturative. Secretory will be basically forming the organic matrix, first the organic matrix, because first the organic matrix has been laid down, and entire girth, or you can say entire height of the entire, what you can say, landmark, or entire width is been taken in consideration at the secretory phase, because first, what you can say, the outline is made ki at this part the enamel is going to form, and it will be restricted to this area. And maturation is just what you can say, maturation is just increasing the content, or you can say increasing the height, length, and width of the enamel rods, whatever has been laid in this layer. So this was about amelogenesis in short. Okay, now we are going to study the life cycle of ameloblast, because studying life cycle of ameloblast is studying amelogenesis. They go hand in hand. So now we are going to study life cycle of ameloblast. Now this life cycle of ameloblast is divided into 5 stages. First is morphogenic stage. Second is formative or secretory phase. Then is your maturative phase. Oh, sorry, I've mistaken over here. First is morphogenic, then is the organizing phase. Third is your formative or secretory. Then is your maturative phase, and then protective. Okay, before discussing the life cycle of ameloblast, we should also know two concepts, that is organizing influence and reciprocal influence. Now this is your internal enamel epithelium, external enamel epithelium. Now this internal enamel epithelium contains pre-ameloblast. These pre-ameloblasts are not been yet differentiated into ameloblast to secrete enamel. Now this pre-ameloblast cells, or you can say internal enamel epithelium, influences in the early bell stage, influences or gives signal to your developing, or you can say undifferentiated ectomesenchymal cells, which are in the dental papillae area. Over here there are undifferentiated ectomesenchymal cell. This ectomesenchymal cell is been signaled by your internal enamel epithelium to differentiate into odontoblast cells. I'll make the odontoblast in red color. They differentiate. This is not the proper odontoblast what I'm making. It's just a reference diagram because odontoblast doesn't look like this. Okay, so this internal enamel entire over here also. This internal enamel epithelium gives signal to your undifferentiated ectomesenchymal cell to form a cell that is called as form a layer of cells that is your odontoblast cells. Now this odontoblast cells are going to lay down your first layer of dentine. It is going to lay down first layer of your dentine. So the signaling which was passed from this internal enamel epithelium to the undifferentiated ectomesenchymal cell to differentiate into your odontoblast cell, this is called as your organizing influence. Now this dentine which has been formed, the first layer of dentine which is in form, when it comes in contact with this ameloblast cell, it gives signals to your ameloblast, or sorry, not ameloblast, the internal enamel epithelium, to differentiate into ameloblast cell and secrete enamel. So this kind of influence is called as a reciprocal influence. So now you're clear about the organizing and reciprocal influence. This is very important, organizing and reciprocal influence, without which the process of amelogenesis or the process of dentinogenesis also is not going to start. And you should also remember that dentine is the first hard structure to be formed of a tooth. Okay. So now we are going to just make a quick review of this all life cycle of ameloblast. Now first is morphogenic. Morphogenic is just you're going to see how the cell is going to look morphologically. It is just low columnar, or you can say cuboidal, where the nucleus is centrally placed with your Golgi bodies at the proximal end, with rough endoplasmic reticulum, very small amount of rough endoplasmic reticulum, and then what you can say mitochondria, which is scattered all over. So this is your pre-ameloblast, or you can say as inner enamel epithelium, the cell of inner enamel epithelium. Now this is just about the morphogenic stage, how morphologically the cell looks like. Now the next stage is your organizing stage. Now in organizing stage, the cell attains some height, it becomes high columnar. Then the nucleus, which was centrally placed, suddenly move at proximal end. Here down there is a basement membrane. It moves upwards. This is your what you can say nucleus. Then the above Golgi apparatus, whatever the Golgi bodies were at the proximal end, they travel downwards due to the shifting of nucleus. Then there is scattered mitochondria, and now the number of what you can say rough endoplasmic reticulum increases because there is now higher activity of protein synthesis. And the basement membrane starts degenerating. At this stage, the first layer of dentine has been laid down by organizing influence. Because the first layer of dentine has been now laid down, now this dentine comes in contact with this cell, and this cell is now fully matured ameloblast, called as your ameloblast cell. Now we are going to see the next stage, that is your formative stage, or you can say it as secretory stage. Now your amelogenesis starts. Exactly, the amelogenesis, the process of amelogenesis starts. Now in formative stage or secretory stage, at the distal part, you'll find some secretory vesicles. Now this secretory vesicles will have your enamel proteins and some of your hydroxyapatite crystals or calcium content, which is going to be laid down above the first layer formed dentine. This is your dentine, and this formed enamel or the enamel matrix, which whatever it is formed first, this enamel is going to be highly inorganic content because it contains only 30% of mineralized content or mineral constituents. So this is about your ameloblast. This is just at the starting stage of your formative starting stage of your life cycle of ameloblast or formative stage. At formative stage you will see that this vesicles merges with the plasma membrane at the distal end and secretes your primary enamel, which is organic, and it also contains 30% mineral contents. So okay, so that was your how enamel was, first layer of enamel is formed. Now as the first layer of enamel is being laid down, suppose this first layer of enamel is been laid down, primary enamel. Now the layer here is the dentine. Now this ameloblast cell shows migration. This ameloblast cell moves upward towards the external or collapsed, you can say, external enamel epithelium, or you can say away from the odontoblast. If this is the odontoblast, this is the odontoblast, and here there will be pulp. This odontoblast, this is the odontoblastic process. It will move away from the odontoblast with the help of, by leaving a process that is called as your Tomes process. So as it will move away from the dentine part or away from the dentine, it is going to form the Tomes process or leave its process that is called as Tomes process. Now Tomes process is actually the cytoplasmic continuation, but it is just a process which is being noticed in the life cycle of ameloblast at this stage. As in forward, like it is moving, it is going to leave its process. Sorry, okay. This also contains secretory vesicle, and it is going to secrete your enamel matrix. It is going to secrete the enamel matrix. So this was your dentine, your first formed enamel matrix, and then it is going to secrete your enamel, the further enamel matrix or primary enamel. So just going into detail of how this has been secreted. It's a blunt process. Tomes process is a blunt process, and it actually resides inside the pit, or it see, now we'll get a clear idea. See, imagine this is your ameloblast. Now when the ameloblast was secreting your first layer of enamel, or you can say primary enamel, just at the starting stage, it was just a layer of, what you can say, a layer of enamel rods which has been laid down. So this kind of enamel is called as your prismless enamel or structureless enamel. Now when this cell is moving away, as well as it is secreting simultaneously the enamel matrix, so you will find something like this. This is the first enamel, ameloblast, the second ameloblast, your third ameloblast. I'm just giving you example. Here there is a vesicle which is going to secrete the other vesicle, then the third vesicle, and this is your Tomes process. Again, at the junction there is a vesicle, junction of your ameloblast and your Tomes process. Then there is again a vesicle. which is secreting your enamel matrix. Okay, now I'm going to show it the rod. Now primarily when it was secreting this enamel, so it was somewhere here now. Now as the cell is moving upward, it is leaving its processes. That process is called as a Tomes process. So this was the part of your enamel which was it's secreting. This was your dentine. Now, as the blunt process is formed, these are also going to secrete your enamel matrix. Now, this orientation of this enamel matrix will be at a particular angle because it's going to secrete like this because there is no other chance of secreting. So, this is going to give you enamel rods at this direction, or it's going to secrete enamel rod or enamel matrix at this particular angulation. And this is straight, so this will secrete in this pattern. So, and this is called as a pit. Okay, this Tomes process is inside the pit. Here will be the adjacent one, again the same case history. Okay, so this is called as your interrod enamel because this is called as your interrod enamel. Whatever like the enamel is secreted by the junctional or at the junction of your ameloblast and your Tomes process, this is called as your interrod enamel. This is called as your enamel rods, and this is actually the enamel which was secreted at the starting stage. This is called as your prismless or structureless enamel because there is no orientation seen in the enamel rod. It is just in a single layer, but over here you can see a specific angulation due to which this both enamel is been clearly distinguished in the what you can say when you see in electron microscope. But though they are been distinguished or they are been considered as two different like when we see it is just like it is two different structures because when you see in electron microscope you will be saying no, this is something else and this is something else, but remember that these two contents are exactly carbon copy 100% same because this is also the enamel matrix secreted by the cell and this is also which is secreted by the same cell, but due to the different angulation of enamel rod they have been differentiated, or you can say they are just said these are the enamel rods or these are the interrod substance, or you can say interrod enamel, and this is going to be your structureless or prismless enamel. So this was something in your secretory or formative stage. Now the next stage is your maturative stage. Maturative stage, in maturative stage there is reduction of your height of your ameloblast. Now this ameloblast show modulation. It will show two types of ameloblast. Now and the Tomes process has been degenerated. Okay, one will be your ruffle bordered and one will be your smooth bordered. Now the layer which now here over downside your enamel is been secreted, your enamel or primary enamel has been laid down. Now this is the second stage of your amelogenesis, that is this is a structureless enamel, for example. Just making a reference diagram, okay? Don't take it exactly. Okay, so this is now your maturative phase. Now this is very important to know that in maturative phase, in this phase, now there will be influx of all your mineral content and all the hydroxyapatite crystals have been laid down, and it has been it undergoes maturation and your enamel becomes 96% mineralized. and the whatever the rest of the water content and organic content is been effluxed or taken out from this enamel matrix, and just a traces of 4% of organic matrix and water has been left over in this enamel matrix. So the ruffled borders, this will execute, or this will help in deposition of minerals. It will deposit your inorganic content or inorganic matrix or inorganic hydroxyapatite crystals, and the smooth borders are nothing but just they are going to absorb, or they're going to efflux the water and your organic content. There is a other theory also, like where you see one of the enamel rod. Now here is the other enamel rod. Now as these enamel rods are increasing in size, the organic content which was in between, the more of organic content which was in between, now this organic content is being squeezed out, because now it is going to increase in size, height, width, and whatever the organic content was there in between is going to be thrown out. It will be taken by this cell. So this is going to take out your water and organic content, and just a small thin trace which we call 4% is being left over. So this was something about maturative phase. And protective phase is the last phase, where you're going to see that the enamel the ameloblast is highly reduced in size. This I'm showing a larger view, but they are highly reduced. The layer, above layer, which was collapsed in advanced bell stage, the layer also 1C, for example, this is your enamel formed. I'll show it by blue. It's formed enamel. Here the dentine is formed. Now as these hard structures are formed, the dental follicle cells they collapse, and they just form a single layer now with the ameloblast, or you can say now the ameloblasts are called as the reduced enamel epithelium. With this the reduced enamel epithelium, the stellate reticulum has been degenerated. Now this external enamel epithelium, this comes in contact with your ameloblast cells or reduced ameloblast cells, and they form, or they function as a single membrane, epithelial membrane, that is called as a reduced enamel epithelium. So this is going to form as your reduced enamel epithelium, which is going to support your tooth, or it will just cover your tooth. If this is just a reference diagram, the tooth developing here, the hard structures forming here, it is just going to form a protective membrane above it, and it will not allow, if it will not allow to, what you can say, it will just protect the developing tooth underlying or developing tissues like enamel and dentine. Because if there is premature loss of this layer, if this layer is lost, now this enamel comes in contact again with the dental follicle cells, and the dental follicle cells may differentiate into cementoblast and secrete cementum, and you can see enamel pearls on the layer of enamel. So this was about the process of amelogenesis and life cycle of ameloblast, which we have discussed in detail. So I hope you have enjoyed the lecture video. So we'll meet soon with other concepts and other interesting topics of dental histology. Thank you. Thanks a lot.